Okay, once again, announcement or two, once again, our lab today is going to be the second of the photosynthesis labs. We'll collect those completed labs for both of them tomorrow. And then we're going to do the first of two electrophoresis labs on Wednesday. Following Monday and Tuesday, we'll do the, we'll put the enzyme lab first, because that way it'll just, that's a fairly involved lab, 2 days worth. And that way we'll get things kind of out of the way, and you guys will have a little bit of time to write that stuff up. And then we'll do a second electrophoresis lab on that Wednesday. Okay, so yes.
I just want to know, what are your lectures called on YouTube?
Oh, if you want to go to, just go for Sauterer cell biology. That will show all of it. It's about 180 things between cells. There will be ones for cell biology and ones for developmental biology. They're all up in the same place. I put the same tags in to make sure that they would all end up in the same place for people to get them. So
do you put the dates on or anything? So
no, I put topics though, and subtopics. Like the last one I put up, I had one, it's called RNA interference, another called L and C RNAs, a third one was amino acids, peptides, and proteins, and then the fourth one was protein structures. So I break the subsections down, and they range from about 10 or 15 minutes to slightly over half an hour, depending on what you want. So, and there's just a few more I'm going to do today, because then we have sort of a complete library. So I'm going to do a couple more of these guys today, post them up, and the rest are already posted. It'll be the same material, just broken up into different things. So we're going to continue on proteins. We looked at protein structure, the primary, secondary, tertiary, and quaternary. Once again, tertiary and quaternary structure are critical for the protein's function. We gave several examples of things where you alter the such as those prion proteins. You have two stable tertiary structures. One of them is almost indestructible and has a tendency of binding to, and occasionally, very rare, they haven't shown this rigorously yet, but this is the widely held hypothesis, that once in a while the disease structure will bind to a normal prion protein, flip it into the disease structure. So you've effectively multiplied, and then that continues on, and finally stuff piles up, clogs up your nerve cells, and the nerve cells die. And then you get holes in your brain, which is not really a good thing. So that's tertiary structure going wrong. Well, let's take a couple other things looking at tertiary structure and quaternary structure related things. Once again, tertiary and quaternary structure, the primary thing that holds them together is going to be hydrophobic interactions, a tendency of the polar amino acids to stay on the outside facing water. The hydrophobic amino acids tend to bury themselves in the interior. Of course, the reverse is true in a membrane protein, because it's going to be in a hydrophobic membrane environment. Okay, and then you have ionic bonds, in other words, between different charged amino acids. They will weakly stick together even in water. And finally, you have occasional hydrogen bonds that are minor contributors to protein structure. So those are things holding tertiary and quaternary structure together. Now, let's take a couple more examples of related protein structure. Let's start out with something. Okay, we're going to start out by taking an egg, cracking open an egg. Okay, you probably did this morning. Crack open an egg. You look at the egg white. The egg white is transparent, sticky, goopy kind of thing, really dense. And what it really is, of course, is a very concentrated solution of proteins, maybe about 30% protein by weight. And the major protein is called ovalbumin or egg albumin. And there's others in there, but the vast majority of this is one protein. So it's such a highly concentrated solution that it turns into almost a sticky, semi-jelly-like thing. Okay, now we crack that egg open and toss it in some boiling water. What happens after a certain period of time? The egg white turns white. And it has a more solid gel-like texture, gelatinous kind of texture. So you take that thing out. You've cooked that egg, and something has happened. Something has happened at the level of the proteins in that egg white. Those proteins are now reflecting and absorbing light differently. Instead of being transparent, now they're reflecting light. That's why it turns white. Its physical characteristics, from a goopy, thick jelly-like solution to something that's more solid. So we've done something to those egg white proteins by throwing in boiling water. Now you take that thing out of boiling water and you notice it does not turn back. What's going on here? So heating those proteins up has done something to those proteins. And that apparently seems to be irreversible. You can hard boil an egg, but has anybody ever been able to unhard boil a hard boiled egg? No. Why? Okay, example number 2. You take a glass of milk, and then you start pouring and stirring in vinegar, lemon juice, any kind of acid, hydrochloric acid, you get whatever, any kind of acid will do. You start stirring the acid into the milk, dropping the pH. And then after a certain point, you start seeing little tiny flecks forming in the milk. And the flecks get bigger and bigger and bigger. And finally, they settle out of the solution. What you have left, what they call whey, is kind of transparent. It's not milky white anymore. It's kind of transparent. It's got stuff in it. But a lot of things have settled down. These clumps of stuff, of kind of gooey stuff. That's what we call curdling milk. Now, what's going on in that? What's happening with that hard-boiled egg? What's happening with that curdled milk? One other example. Let's suppose you're going to slice up some potatoes and fry them or, roast them or whatever the case is. You make a whole bunch of potato slices and then you get waylaid. Maybe somebody comes over or something like that. So now you got all these potato slices sitting on the countertop and stuff. And after a while they start turning funny colors on you. They start turning red and brown and stuff like that. Well, the reason they're turning funny colors on you is there's an enzyme. In fact, that's what the enzyme we're going to be working with the enzyme lab. There's an enzyme that does a number of reactions on similar substrates, and it turns what are generally colorless substrates into substrates that are orange, brown, even black. And apples will do that, and mushrooms. That's why mushrooms turn black on you after a while. Now, on the other hand, if you boil that potato first, and then cut it into slices, you could leave it outside on the plate until the thing started to rot on you, and it would not turn funny colors. So all 3 of these are examples of doing things to proteins. In this case, in all 3 cases, by either extremes of pH, adding acid to milk, or by extremes of heat, throwing something in boiling water. What have we done here? Well, this is what we call denaturing a protein. Now, what happens when you denature a protein? What you do is take that beautiful tertiary quaternary structure and you disrupt it. You break those weak hydrophobic interactions, those weak ionic bonds, those weak hydrogen bonds. Boiling water is enough to do that to almost all proteins. And when you break all those bonds that are holding tertiary and quaternary structure together, the protein unfolds into a floppy chain of amino acids. So you unfold the tertiary structure. And of course, quaternary structure as well. So you go from here, nice tertiary structure, and then now you go back to a floppy chain of amino acids. Now the act of denaturing proteins, just these examples I gave you, illustrates a few things about what happens when you denature proteins. One of them, for the most part, you denature proteins, it's a one-way ticket. You don't unhard boil eggs. If you curdle milk by adding acid into it, removing the acid and bringing it back to normal pH does not uncurdle the milk. You still got that stuff at the bottom, which is the milk proteins. And a second thing we've seen here with the potato example, or you could do that with an apple or whatever, is when you boil those guys, that enzyme no longer worked. That's why the potato, a boiled potato doesn't turn funny colors and a raw potato does. You have killed that enzyme's biological activity by denaturing it. So, we see a couple features when you denature proteins. We'll see why in a little bit. It's usually irreversible. It's a one-way ticket most of the time. We'll see a few exceptions. Okay, it kills off the biological activity of the protein, at least using that enzyme as an example. We saw from the egg white that it's going to alter, in many cases, the way the protein interacts with light. Now, what's going on here? Okay, I'll give you an example using curdled milk. We take our milk proteins, right? Major milk protein called casein that's often used as a protein supplement that you get in these health food stores and stuff like that. Milk protein, curdled milk basically, and they sell it for a fortune. Okay, so here's your normal tertiary structure. Now we denature it. Okay, the tertiary structure unfolds. You got a floppy chain of amino acids. Now here's the problem. Here's why curds form, and here's why you cannot uncurdle milk or cannot unhard-boil a hard-boiled egg. Recall, protein structure is largely held together by the hydrophobic interactions. The hydrophobic amino acids try to stay out water. The protein tries to fold up to keep the hydrophobic stuff away from water. So now we got this protein, and let's say here's a hydrophobic region. I'll put H down there. Okay, when the protein's folded up, that region is buried in the inside of the protein, away from water. Now we have that exposed. That hydrophobic region does not want to be in water. It wants to get out of a polar environment as quickly as possible. However that happens, you'll want to get it out. Okay. Alexandra, let me get this to you. All right. Okay. So, there's two ways this molecule could go back and resolve this little situation. One of them could simply be to refold. Go back into its original tertiary structure. The other way would be just to clump together with another one of these proteins. In fact, continue to clump.
I thought it was irreversible.
Now we're going to see, yeah.
Sometimes.
Well, usually, I'll mention a couple. These are possible pathways. Okay, so now we have one denatured protein. Along comes another, and the two hydrophobic regions stick together. So we partially shield them. Along comes yet another, and we shield it a little more, and then comes another, and so on. You keep on doing this, forming bigger and bigger and bigger clumps. And the bigger the clumps are, the more hydrophobic stuff is shielded from water. Now, once a protein has done that, the major thing, the hydrophobic interactions have resolved themselves. You have now shielded the vast majority of the hydrophobic stuff away from water. It's buried in the inside of the clump now. At that point, the protein doesn't have any tendency to refold. It's already happy. Fortunately, it doesn't work, but it's already happy. So, remove the denaturing agent, cool the thing down, get rid of the acid, whatever the case is. The protein's still not going to be likely to fold up. So these are the two possible pathways. You can refold or you can clump. Problem is, protein folding takes time. Clumping is a lot faster. So, hey, if you got hydrophobic stuff exposed to water, you want to do this as quickly as possible. So generally, you will clump faster than you can refold. And that, folks, is why most of the time you denature a protein, the protein clumps together irreversibly. There are a few rare exceptions, generally very small proteins. There are some proteins that will actually fold up faster than they can clump. So you can denature these proteins, remove the denaturing agent, they fold back up again, and act normally like nothing ever happened. But that's really the exception rather than rule. And that only applies to small proteins with molecular weights like 10, 20,000 or so. And not all of them either. Big proteins are almost always irreversible once you denature them. Okay, now, we see this case with Mr. Carpenter. Okay, we see this case with denatured proteins. Now, what kind of things can denature proteins? Well, obviously, we see a number of things that can do that. We've already seen some. Heat will do that. Now, a lot of proteins, you know, we use the examples of bringing things up to boiling hot temperatures. They'll denature almost all proteins, not every one of them, but it'll denature the vast majority of proteins. But most of the time, you don't have to heat things up even that much. A lot of proteins will denature at temperatures no greater than that at a hot summer day in Alabama. And that's one reason, folks, why most organisms cannot stand high body temperatures. If you get a fever, you start, you know, feeling a little off, and you stick a thermometer in your mouth, it's 100 or 101 degrees Fahrenheit. Okay, you start feeling kind of crappy. You start feeling worse, maybe it's 103, maybe even 104. By the time you get to 105, you're feeling pretty darn miserable. And you got to think about going to the ER. If it starts going up to 107, 108 degrees, you are in danger of your life. And they will probably put you in a cold water or even an ice bath. You know, you hear lots of times football players, especially practicing in the summer and stuff, dying of heat stroke because their core body temperature's gone up 107, 108 degrees. They collapse on the field. They're dead by the time the EMT people get there. What's going on there? And if you happen to be a cold-blooded or ectothermic organism, like a reptile, you ever notice, especially this time of year, you drive down the road in the morning and you see things like snakes and turtles and all these kind of lizards sunning themselves on the road. They're trying to warm themselves up, raise their body temperature to the point they're active enough that they can go around foraging and avoiding predators and things like that. But on a very hot day, especially in desert environments, you're not going to see these guys around. What they do is they hide. They burrow, whatever the case is, because otherwise their body temperature can risk going too high, and that will kill them. How come? Because even at temperatures as little as like 106, 108 degrees Fahrenheit, that is enough to denature numerous proteins, especially ones in your central nervous system. So you literally cook your brain. You start denaturing proteins that are necessary for nerve cell function. You go into a coma, and then you're dead. So it doesn't take extremes of heat to denature many proteins. Boiling water will do it to almost all of them, but many proteins denature at temperatures not much greater than that of a warm bath. Okay. As a matter of fact, speaking of that, there's actually some warnings that people who have hot tubs should not set them more than about 102 or 103 degrees Fahrenheit, because you get somebody have it like 105, like a warm bath, a fairly warm bath, and they're in there relaxing and stuff, and they fall asleep or something. They can actually raise their body temperatures dangerously high levels. There was cases of people that actually killed themselves in their own hot tubs inadvertently because it warmed their core body temperature up to dangerous or even lethal degrees. So that can happen. Okay, so heat can do that. Now, interestingly enough, different proteins have different heat tolerances, and different organisms have different heat tolerances. For instance, there are organisms that are called hyperthermophiles, meaning they love extremely high temperatures. Those include certain types of bacteria and certain types of archaeans. These guys, as long as water stays liquid, some of them can actually live and grow at temperatures above the boiling point of water. They're found in deep sea volcanic vents and things like that, where the tremendous pressure of the water raises the boiling point to higher than oven temperatures. But they have grown, some organisms, successfully at 120 degrees Celsius. That's as hot as an autoclave. And some of these organisms can not only survive, but can actually grow under those conditions. Now, how do they prevent their proteins from being denatured? You look at the structures of those and the sequences and compare them to the normal version. Many times they have lots and lots and lots of charged amino acids, and you get lots and lots and lots of ionic bonds that help to stabilize the protein's tertiary structure at high temperatures. So they have tricks. They modify their proteins so they don't denature at even temperatures at or even somewhat above the boiling point. But they have special proteins, special adaptations to hold their proteins together at these very high temperatures. Okay, so heat can obviously do it. We said extremes of pH can do that. That's one reason that very few organisms can live in either very acidic or very basic environments. Most organisms get into serious trouble if they're exposed to environments less than about pH 6 or pH 5 in some cases, or more than about pH 8.5 or 9. There are some organisms here, again mostly microorganisms, that can live at fairly extreme pH. And here again, reason why is many proteins denature when you get out of a relatively narrow range of pH. Like you go below about 5 or above 9 or something, and quite a few proteins denature unless they've been specially designed to tolerate those pH extremes. So once again, there are not too many organisms that can live in extremely acidic or extremely basic environments, and they have special adaptations to deal with it. So pH can do, pH extremes can denature proteins. Other things that can do it, detergents. Your typical detergent molecule is a hydrophobic molecule, basically a hydrocarbon chain, and frequently they have some kind of ion at one end of it. So your common things that you use to wash your dishes with, wash your clothes with, wash your face with it, they're basically fairly hydrophobic molecules, often with a polar or charged end on it. In a sense, not too dissimilar to what we're talking about, some of these membrane lipids and things like that. Okay, now what happens if you have a proteins and you throw them in an environment full of detergent molecules that are hydrophobic, the protein polar surface is going to be exposed to these hydrophobic molecules. They don't like it, so they want to get the hydrophobic stuff out, so the protein tries to flip itself inside out and denatures in the process. Now, why do you use that for washing clothes? Let's suppose you got some protein stain, blood stains, meat juice, or whatever on your nice cotton shirt. Your cotton shirt is made of cellulose. It's a very polar molecule. The polar proteins in the meat juice or gravy or whatever, blood, whatever the case is, those are going to stick nice and tightly to that very polar surface of the cellulose in your cotton. Now, you throw it in the washing machine, throw in some detergent. The proteins end up denaturing themselves. They no longer stick to the polar cotton. They go into these bubbles of detergent, and then they go out the drain and down the sink. You've just got these protein stains off of your clothes. That's how detergents work. And of course, it also will dissolve greasy stuff like the oil that you spilled on your shirt or pants or whatever the case is. So, detergents do that. And organic solvents do that. In other words, hydrophobic organic molecules. Now, for instance, that's one reason why you do not want to drink gasoline if you're siphoning gas out of your car or hopefully not somebody else's, because gasoline is very hydrophobic. It's a bunch of hydrocarbons. You gulp the stuff down by mistake, bad idea. As it goes down, what's going to happen? It's going to start dissolving cell membranes, rupturing the cells, and it will also denature the proteins it's exposed to. So they actually tell people if you actually swallow gasoline, do not induce vomiting because it's going to do as much damage coming back up as it did going down the first place. Okay, don't siphon gasoline by mouth. That would be very sick that way. But organic solvents partially work as well by denaturing proteins. Okay, so that's what we see when we take an extreme thing of denaturing a protein, completely disrupting the protein's tertiary and quaternary structure. And once again, the vast majority of times the proteins will clump rather than fold back up again. But there are things that will affect protein structure. Denaturing a protein completely disrupts the protein structure. But you can alter the protein structure by these kinds of agents without necessarily going all the way to denaturation. Now let's give an example. In fact, we're going to do this in one of our labs. Let's take an enzyme of some kind, whatever kind. And we have some kind of measure of its activity, whatever it does. And then we plot that over pH. We're going to make a pH activity curve for an enzyme. So let's say 3, 4, 5, 6, 7, 8, 9, and God forbid 10. Okay. Now, most of our enzymes happen to work best at a pH around neutral or maybe a little bit basic, which is your body fluids. Body fluids in the insides of cells tend to be about pH 7.2 or so. So, using a typical kind of enzyme, you'd have peak activity at a little bit more than 7, something in that general range. Now, if we start changing the pH, when we're changing pH, of course, what we're doing is altering the concentration of hydrogen ions or and OH ions in the solution. These ions, of course, are charged. And what these ions will do in high concentration, they'll cluster around polar and charged molecules in the proteins, and they can start moving them, pushing them in, pushing them out, whatever. In other words, pH, by the attraction of these ions, these various amino acids, pH differences can alter a protein's tertiary structure. And if you do that with an enzyme, for instance, you'll alter the structure of the active site, the catalytic part of the enzyme. And if you alter it, probably it's not going to work as well as it did before. So what happens? You'll see the enzyme activity drop off outside a certain pH, optimal pH thing, kind of like a bell curve thing. And we'll go down and down. It can get really, really low or virtually non-existent. And then you reach a certain point, and bang, you have denatured the protein. You took it one step too far, now you've denatured the protein. You've probably killed it off irreversibly. Okay. Now, different enzymes, of course, have different pH optima. For instance, look at 2 protein-digesting enzymes that are found in our own body. The major enzyme that digests proteins in our intestines is known as trypsin. It's produced in the pancreas and secreted into our intestines. Now, if you stuck a pH meter in your intestines, which would probably hurt quite a bit, you would find the pH is about 8, maybe 8.2. It's a little bit on the alkaline side, a little bit on the basic side. Lo and behold, the enzyme trypsin, lo and behold, works best at about pH 8. It works not quite as well at 7, and certainly not as well as at 9 and stuff. Now, on the other hand, you go into your stomach. Your stomach, you have cells that are pumping hydrochloric acid into it. So the pH of your stomach is about 1.5. It's quite acidic. There's a protein-digesting enzyme in there, too, called pepsin. And guess what? That enzyme has a pH optimum of around 2. At neutral pH, it won't work at all. If you take trypsin and expose it to pH 2, you're probably going to denature it. So we have a protein-digesting enzyme in our stomach that works best at extremely low pH. Another one in our intestines that works best at slightly alkaline pH. And you put one in the other, it either won't work at all or will flat out denature. Not only that, the acid in your stomach has a tendency of hydrolyzing the peptide bonds in and of itself, and then it gets helped along by these enzymes. And of course, we all know, stomach acid is quite corrosive. If anybody has acid reflux, chronic heartburn, in other words, what happens? Some of that stuff leaks out and starts burning your esophagus. If you've ever had the misfortune of throwing up, you often feel that burning sensation as all that stuff from your stomach and all the acid stuff comes out up your esophagus, out your mouth, out your nose at times. That is, oh God, tell me, that is nasty. Okay. All right. And it burns because it's got lots of hydrochloric acid in it. So it makes it a totally miserable experience. Okay. So we have these changes in pH. Now, it turns out in certain kinds of cellular organelles, we deliberately keep the insides of those organelles rather acidic, and the reason why is the acid pH actually inactivates the enzymes inside. And only when we either get those enzymes out of that organelle or raise the pH in that organelle do those enzymes wake up and start doing their work. So sometimes in cells we actually use pH changes in local organelles or membrane-bound compartments, and we keep them at certain pHs, do things like inactivate certain enzymes, whatever the case is. Okay, so we can see that We can alter enzyme activity, and this is a general thing for protein. We can alter protein activity, function, stuff by changing the pH. Likewise, if we were to do things like salt, we're going to do that with our enzyme lab. Okay, if we start doing things like that with salts, once again, just about all kinds of proteins will work well at certain salt concentrations and not work well at others. Many of our enzymes and other proteins work best at about 0.1 molar salt, which is about what we see inside cellular fluids and body fluids and things like that. If you put it in really low salt, some enzymes and some other things don't work at all. If you put it in really high salt, same kind of thing. And here again, when you're adding in salts, what's happening? You're adding in all these charged ions and they can alter protein tertiary structure. And that will alter the protein function. So we see, by these examples, protein tertiary structure is actually quite fragile. It doesn't take much. A little bit of heat, a little bit of detergents, too much acid, too much base to outright kill a protein, to denature it.